Today we are going to learn about the cDNA libraries. cDNA, it is a complementary DNA, and that complementary DNA, it is going to be synthesized from the mRNA as an template. So from a cell, the mRNA, it is extracted, and that mRNA now it is converted into the cDNA, and the total collection of that cDNA, it is going to be called as the cDNA libraries. Now, these cDNA libraries, it has the advantages over the genomic library, as the size of the cDNA clone, it is a smaller than the genomic clone. Here, the number of vector molecules which are required for the cloning purpose, it is a lesser than the genomic library. Second, when there is a synthesis of the cDNA, the template which is going to be used, it is the mRNA. And when there is a synthesis of the mRNA in the eukaryotes, okay, when there is a maturation of the mRNA, at that time, the introns which are present into the nascent mRNA, it is going to be removed. So the mature mRNA, it is having only the exons, and now total exons, now they are going to be converted into the cDNA. So the cDNA, it is not consisting of any introns. Now such a type of an cDNA, it can be cloned into the bacteria. Okay, it can be cloned into the bacteria as it can be cloned into the bacteria as it does not consisting of any intron. So the eukaryotic gene, it can be it can be expressed into the bacteria by using the cDNA. Okay. Now the next year, when we construct the cDNA library, that cDNA library, it is going to be a representative of an cell type, or it is going to represent a particular developmental stage of the organism. Now, when we go for the isolation of the mRNA, we select a particular type of an cell, and now we know that in the eukaryotes, in the eukaryotes, there is a tissue specific expression. So here the genes which are going to be expressed into the different types of the cell, they are going to be a different one. That's why here that particular cDNA library, it is going to represent a particular type of an cell. Like how here when we take the pancreas, and in the pancreas the two cells, the beta cells and the alpha cells, these two cells, it will have the different type of the cDNA library, because the expression pattern of these two cells, it is going to be a different. Now the next one here, it is what? And the cDNA library will also be enriched for the abundant mRNA, and it will represent the fewer clones of the rare mRNA. Now, in the cell, there are the genes which are high, which are expressed at a high amount. Okay, that is expression level, it is a high. Some they are moderately expressed, and some they are the rarely expressed. So when we go for the extraction of the mRNA, and when we make the cDNA, here, the abundant mRNA, it will give the abundant amount of the cDNA. Rare mRNA, it will give the rare amount of the cDNA, and the moderate, it will give the moderate amount of the cDNA. So when we construct the cDNA library, it is going to represent more of the mRNAs which are abundantly present into that particular type of an cell, and the rare mRNA represented, they are represented at a lower level. Next year, it is what? That cDNA library, it is also going to represent the alternative splicing variants. Now, here, the mechanism, it is called as an alternate splicing. It is a post-transcriptional modification, and in that post-transcriptional modification, in the splicing, one of the type of the splicing, it is alternative splicing. Okay, that is from one gene. Okay, from the one gene, the mRNA which is going to be synthesized, that mRNA, now it is going to be spliced in the two alternative ways, which is going to give you the two different types of the proteins. So here, this cDNA library, it is going to, it is also going to represent the alternative splicing variants, which are not going to be observed or observed into the genomic library. Now, here, when we have to go for the construction of the cDNA library, the major steps, they are what? The first, it is a first strand DNA synthesis. Okay, second one, it is a removal of the RNA template. Third, it is a second strand DNA synthesis. And the last one, it is a cloning cDNA in a vector. So going towards the first one, it is obtaining the cDNA. There are the different strategies by which we can synthesize the cDNA. The first one which has been given, it is a Maniatis strategy. Then there is an improved method, Gabler-Hoffman method. Then there is a capture method of the full length cDNA cloning, and next one, it is a oligo capping. Going towards the first one, and it is a Maniatis strategy. Now here, when you look at the mRNA, okay, this it is an mRNA, and when you look at this mRNA, and the basic structure of the mRNA, it is what? It has a 5' cap and it has a 3' polyadenyl 10, poly A 10. Now, when we go for the extraction of the mRNA, total mRNA, it is now present with you. And here you have to okay, you have to make the cDNA from all types of the mRNA. So, all types of the mRNA, it will have the basic common feature, and that basic common feature, it is what? It is the poly A 10. Okay, so the known part of the mRNA, it is a poly A 10, so that we can design a primer, and that primer, it is a oligo-DT primer. That oligo-DT primer, now it can anneal with whom? It can anneal with the poly A 10. Now here, when we have to synthesize the first strand of the cDNA, at that time here you are going to use the enzyme, and that enzyme, it is a reverse transcriptase. Reverse transcriptase, it is a RNA-dependent DNA polymerases. That is, it is going to use the mRNA as a template for the synthesis of the DNA. This reverse transcriptase, now it is allowing the synthesis of the DNA, that is the first strand of the cDNA, and here there is a use of an alkali, so that alkali, now it is allowing the removal of the mRNA from the first strand of the cDNA. Now, when there has to be the synthesis of the second strand of the cDNA, at that time there is a need of the primer. And here you can see, when there is a synthesis of the first strand of the cDNA, there is a loop structure, it is going to be formed. And now this loop structure, it is going to be used as a primer for the synthesis of the second strand of the cDNA. So here the enzyme which is used for the polymerization, it is a DNA polymerase. So in this way, there is a synthesis of the second strand of the cDNA. But now when you look at this structure, it is a hairpin structure. So such a type of an hairpin structure, it is not required. And for that purpose, there is a use of the nucleases, and it is a single strand specific S1 nucleases. This S1 nucleases, now it is going to glue this loop structure, which is a single stranded one, and here you can get a double stranded cDNA. Okay, now for the cloning purpose, okay, when we have to clone this cDNA into a vector molecule, okay, to generate the complementary ends, there is a use of the terminal transferases, and this terminal transferases, it is going to add the polycetal. If you are using the DCTP, it is going to add the polycetal at the 3' end. And then on the vector also, you are allowing the terminal transferases to act, which is going to synthesize the poly G10. So here there will be what? There will be the when the vector molecule and your cDNA, it is a mixed up, then there is what? There is the annealing of this cDNA with the vector molecule. Okay, in this way, the cloning, that is the directional cloning, it can be done. Now the second method, it is the improved method. Now in this method, when we are using the S1 nuclease in the Maniatis strategy, when there is a use of the S1 nucleases, some of the information, it is going to be lost. How? When? Okay, when there is a cleavage of this loop structure, at that time the S1 nucleases, it is also able to cleave the some double-stranded DNA. So there is some loss of the information. So to recover that, okay, we don't want any loss of the information. That's why the method, it has been improved. And now in this method, as usual, there is a use of the oligo-DT primer, which is used for the synthesis of the first strand of the cDNA. Okay, then here the enzyme terminal transferases, it is used which is going to add, okay, which is going to add the poly C tail, okay, which is going to add the poly C tail at the 3' end when there is a use of the DCTP. Okay, now here the 3' end, now it is what it is consisting of? It is consisting of the poly C. That is the at the 3' end there is what? There is a known part. Okay, now here the mRNA, now it is going to be hydrolyzed, okay, and there is a recovering of the full length of the cDNA. Now when you look at the structure of this cDNA, what you find out at the 5' end there is a poly T, and at the 3' there is a poly C tail. So both of the ends now they are not known to you. So here when you have to synthesize the second strand of the cDNA, okay, you can design the primer for this poly C. Yes, you can design the primer for the poly C as an what? As an oligo DG. Okay, so here there is, okay, here there is a use of whom the reverse transcriptase enzyme, which is allowing the synthesis of the second strand of the cDNA. Okay, now the next method, it is a Gabler and Hoffman method. Okay, in this method, here it is an mRNA. There is a designing of the oligo DT primer for the first strand synthesis of the cDNA. So here there is the use of the reverse transcriptase, which is allowing the synthesis of the first strand of the cDNA. Then RNase H, it is used to remove the, okay, to remove the mRNA from the DNA RNA hybrid. Then here the scientists they have used, okay, they have used the these primers, okay, and these primers they are called as an random primers, which are allowing the full length cDNA synthesis. Okay, in this way, you can get the duplex of the cDNA. Now, the next method, it is what? It is a full-length cDNA synthesis. In now, when you are allowing the synthesis of the cDNA, it may happen that there is an incomplete synthesis of the cDNA. It may be, okay, incomplete synthesis of the cDNA, it may be due to it may be due to the enzyme which is going to be used during the reaction. Second, here it is what that your reverse transcriptase, it is not going to reach up to the 5' end, and that's why there is a no full length cDNA synthesis. Third one, it is what the when you are isolating the mRNA, the length of the mRNA which is isolated, or the mRNA which is isolated, it is not a full length mRNA. So to overcome all these problems, okay, there are the methods they are used here. And of course, the problem of the enzyme, it is allowed to allowed to solve by engineering, okay, by engineering the, that is by modifying the reverse transcriptase. Now here the murine reverse transcriptases, okay, which don't have the RNase H activity. Now when there is a synthesis of the first strand of the cDNA with the help of the reverse transcriptases, they have inbuilt RNase H activity. So here what may happen, the template it is going to be cleaved by the reverse transcriptase itself, and due to that there is no full length cDNA synthesis. Second, it is what? At these reverse transcriptases, they have the optimum temperature of the 37°C. So the mRNA it can form, okay, the mRNA it can form what? It can form this such type of an secondary structures. Now when the enzyme, okay, when the enzyme it is coming at this secondary structure, it is going to leave the site. So the next here the enzyme it is not going to move forward when there is a secondary structure. So here, okay, so here to solve this problem, the reverse transcriptases they are engineered in a such a way that it don't have the RNase H activity, and the second, the optimum temperature of the reverse transcriptases it is shifted from the 37°C to the 50°C. So at the 50°C, no secondary structures they are going to be formed. And as there is a no RNase H activity, the template it is not going to be cleaved by the enzyme. Okay. Now the next one, it is a capture method. Okay, now here it is your, this it is your mRNA, okay, and this mRNA, it has a basic structure and it is having what? It has a 5 prime cap and it has a poly A 10. Now, here, the first strand of the cDNA, it is going to be synthesized. The oligo-DG primer, it is designed with the help of the reverse transcriptase. The first strand, it is going to be synthesized. Okay. And now what may happen? Okay. What may happen that the cDNA, it is going to, okay, cDNA, it is going to be synthesized up to the gap. That is full length cDNA synthesis, it is there. Or what may happen that there is a partial length cDNA synthesis. That is the cDNA, it is not reaching, okay? It is not synthesized up to the 5 prime end. So this one, it is a partial and this one, it is a full length. Okay, so when there is a synthesis of the full length cDNA, at that time there is a no presence of the single stranded RNA. But when there is a partial synthesis of the cDNA, okay, at that time you can see here, okay, you can see here it is a single stranded RNA. Okay. Now in your reaction mixture, both types of the cDNA they are present. One it is a full length and one it is a partial length. So from this you have to pick up this full length. Okay. And for that purpose you are allowing, okay, you are allowing treat your sample with the RNase A. Okay, RNase A, it is an RNase which is going to cleave the single-stranded RNA. Okay, so this RNase A, it is going to cleave the RNA which is a single-stranded, and where the single-stranded RNA it is present in the partial length cDNA, the single-stranded RNA it is present. So here there is a cleavage. Now this DNA RNA hybrid, okay, the partially synthesized cDNA to this DNA RNA hybrid, there is no presence of the cap. Okay, but here when you look at this part where it is a full length cDNA, here what you can see that the RNase A, it don't have the template. Okay, so it is not allowing any cleavage into the RNA. Now the second here it is what that here this EIF4E, it is a initiation factor in the eukaryotes for the translation, and this EIF4E has ability to get bound with the 5 prime end where there is a presence of the cap. So here this property of this EIF4E, it is used for the isolating the full length cDNA. So here there is a preparation of the affinity column, and this affinity column it is made up of, okay, this affinity column it is made up of this EIF4E. Okay, so the affinity column it has been made, your sample it is run through the column, and now what will happen? Okay, here it is what? Here it is your EIF4E, and it has affinity for whom? It has affinity for the 5 prime cap. So here what will happen? There is an interaction between, okay, there is an interaction between the 5 prime cap and your initiation factor. So the full length cDNA now they are going to bound with whom? They are bound with the affinity column. And here you are going to do the washing, and during the washing, these partially synthesized cDNA, they are going to be washed out. Why? Because they don't have any cap. Okay, next here there is what? There is a, after washing the column, then there is a elution of the full length cDNA, and that full length cDNA now you can use for the cloning purpose. Next method, it is a oligocapping. Okay, now here, when there is extraction of the mRNA from the cell, you can get the full length mRNA or you can also get the partial length mRNA. So when it is a partial length mRNA, it has a phosphate at the 5 prime end, and when it is a full length, it has a cap. Cap means what? Methylated guanine, it is present at where? At the 5 prime end. Okay. Now here first there is a treatment with whom? There is a treatment with the BAP. That it is what? It is bacterial alkaline phosphatases. It is a phosphatases means what? It is able to remove the phosphate group. So here, okay, so here there is a removal of the phosphate group from the partial length mRNA. The cap it is going to be retained there. Next there is a use of the tap, and this tap it is going to remove the cap. Okay, it is going to remove the cap from whom? Cap from the mRNA. Now, who is consisting of the cap? The full length mRNA, it is consisting of the cap. So that when there is a treatment with the tap, there is a removal of the cap. Now, here there are the two types of the mRNA that you can find. One which is having the 5' phosphate at the 5' end, and one which is having the 5' OH. Okay, both of them it has a 3' and 5' end, but here at the 5' end, full length mRNA it will have the phosphate group, and the partially length it will have the 5' OH. Now here next one, it is what? There is addition of the oligo cap to the mRNA. Okay, the oligo cap it is added. Now these oligos, now they are able to get bound. That is, it can form the phosphodiester bond only with whom? Only with whom? Only with the which is having whom? Which is having this phosphate group at the 5' end. But here the partial length it has a OH. That's why the oligo cap now it is not able to get bound with whom? The partial length. So the oligo cap mRNA, okay, the oligo cap mRNA now they are allowed to get ligate. Oligo cap mRNA is allowed to get ligate with the 5' end where there is a presence of the phosphate. So oligo capping only it is going to be done to whom? The full length mRNA. Okay, so here now such type of an mRNA, now this mixture of the mRNA it is allowed to synthesize the first strand of the cDNA. Next year, the second end now it is known to you. It is having what? It is having the oligo cap. So you can design a primer for the oligo cap. Yes, you can design the primer for the oligo cap. And now, okay, and now here you will allow the PCR. Okay, that is the sample. It is taken into the PCR, and here there will be the amplification of the cDNA. In this way, there is okay. There is what? There is a synthesis of the full length cDNA. Now, next you have got the cDNA. Now, that cDNA now you have to clone into the vectors. Okay, and the strategy which is used, it is the homopolymer tailing. There is a use of the linkers, and the strategy which has been given by the Okama and Birch. First, going towards the homopolymeric tailing. In this, okay, this it is a here it is an vector. It is a PBr322 vector which has been digested with the PST1. Now here you have the ends which is having the PST1 specific ends. Then there is a use of the terminal transferases which with the DGTP. So as you are using the DGTP, the homopolymer tail it is of the poly G. Okay. Now, as we have seen in the manual this approach, here we have used the mRNA oligo-TT primer, and then we have synthesized the first strand of the cDNA, and then we have synthesized the second strand of the cDNA, and then here there was the use of the terminal transferases with the DCTP. So here there is the ends which are of whom? Which are of the poly C. Now you can see here, this it is a PBr322. It has a poly G ends, and it has a, and your cDNA it has a poly C tail. Now here, these two they can anneal when they are placed together. As the G it is complementary base pairing with the C. Okay, here there is an annealing between the cDNA and the vector, and such type of an vector now it is transformed into the bacterial cell, into the E. coli cell, and in the E. coli cell itself there is a host repairing, and this gap, okay, this gap now it is going to be filled up. In this way, there is a cloning, and this cloning it is called as an directional cloning, because you are giving the direction to the cDNA where to get a needle. Okay. Second strategy, it is a use of the linkers. Now here As we have seen in the Maniatis approach, there is a synthesis of the second strand of the cDNA. And after synthesis of the second strand of the cDNA, here you are allowing the linker. Okay. And here the linker which is used, okay, the linker which is used, it is a Salvan linker. Okay. And this Salvan linker, it is allowed to link to the your cDNA. Okay. Now, linker means what? Linker, it is a small nucleotide sequence which is consisting of the restriction site, that it is called as an linker. So at one end there is a Salvan linker, and at second end there is a use of the EcoR1 linker. That is, there is a ligation of the Salvan and there is a ligation of the EcoR1 linkers at both of the end at the two ends of the DNA. Now there is a restriction digestion with the help of the Salvan and the EcoR1. Now here you can see the ends, they are generated. Okay, the cohesive ends, they are generated. Now when you have to clone this cDNA into a vector, the vector, it has also to be digested with the Salvan and EcoR1, so that there is a generation of the complementary ends on the vectors. So as the ends, they are compatible ends. Okay, there is what? There is the annealing, and then there is what? Then there is a cloning of your cDNA into a vector. Now, next one, again, the next example, here it is of the linkers, how the linkers they are used. So here there is what? Here it is an your mRNA, and here there is a use of an oligo-TT primer, and that oligo-TT primer, it is having, okay, it is having the small piece of the nucleotide which is consisting of the Salvan side, that is the linker. Okay, there is a linker which is attached to whom? Which is attached to the oligo DT. So when there is extension of the first strand of the cDNA, at that time the linker itself, it is added to whom? It is added to the first strand of the cDNA. Next year, the random primers, they are used. The random primers, they are allowing the synthesis of the second strand of the cDNA. Then there is a addition or ligation of the second linker, and it is a EcoR1 linker. Now on the both of the ends, who is present? There is a presence of the EcoR1 linker. Here it is a Salvan linker which has been previously added, and here it is a EcoR1 linker. Now when you are going to do the restriction digestion with the help of the Salvan and EcoR1, you are getting the ends generated for the Salvan and the EcoR1. Now here again, like the previous one, we have to digest the vector with the Salvan and EcoR1, so that the compatible ends, they are generated. And now there will be the annealing of the cDNA with the vector molecule. In the next approach, there is a direct synthesis. Okay, there is a direct synthesis of the cDNA on the vector itself. Okay, so here it is an mRNA. Okay, and mRNA, it has what? It has a 3' poly A tail. Okay, so here the vector, it has been cleaved, and on the vector, with the help of the terminal transferases and the use of the DTTP, there is a synthesis of the oligo. There is a synthesis of the poly T tail on whom? On the vector. Now this vector, okay, and this mRNA, they has the complementary sequences, so that here there will be what? There will be the annealing of the, okay, there will be the annealing of the poly A tail with the poly A tail with who? With the poly T tail. Okay, and then with the help of the enzyme reverse transcriptase, there will be the synthesis of the first strand of the cDNA. Next year, when you have to synthesize the second strand of the cDNA, the known part, it should be present over the cDNA. That's why there is a synthesis of, okay, there is a synthesis of the oligo DC tail. Okay, there is a synthesis of the oligo-DC tail. Again, the same enzyme, terminal transferases, now it is going to be used. But when you are using the terminal transferases, that terminal transferases, it is going to add the poly-C tail on both of them, on the cDNA, first strand of the cDNA, and as well as on the vector. But on the vector, we don't want, okay, we don't want this poly-C. Okay, so here, okay, there is a hint 3 digestion. So here there is a hint 3 site, it is present, there is a hint 3 digestion, so that this part of the vector, now it is going to be removed. Okay, this part it is removed. Well, this one, okay, this part it is going to be removed. Next here, okay, next here there is, okay, there is a use of, okay, there is a use of now whom adapter. And this adapter, it is a DNA sequence which is digested with a particular restriction enzyme. And this adapter, it is a hint 3 adapter. Means what? It is having the ends, okay, it is having the ends which is of having the hint. That is the nucleotide sequence. It is cut with whom? The hint 3, and it has generated the hint 3 ends. And after generation of the hint 3 ends, okay, after generation of the hint 3 ends, with the help of the enzyme, there is a addition of this. Okay, here there is a addition of the oligo G tel, or there is a guanine, that is a D G, they are added. Okay, DG, they are added to the hint 3. Okay, and this hint 3 adapter, now it is allowed to ligate with the vector. Okay, now here, okay, now here there is a complementary base pairing. Okay, this complementary base pairing between the first strand of the cDNA and the vector molecule. Next here, okay, there is a replacement of the RNA strand, okay, by the DNA with the eco R1 RNase H. That is the RNase H, it is going to cleave the RNA from the DNA RNA hybrid, and then DNA polymerase 1, now it is allowing the synthesis of the second strand of the cDNA, and the DNA ligase, it is going to seal the nick. In this way, there is a synthesis of the second strand of the cDNA, and in this way the cDNA, it is going to be synthesized over the vector, and at the same time, it is going to be cloned, okay, into a vector molecule. Okay. Now, which are the vectors then they are going to be used to construct the cDNA libraries? It is particularly here, it is a lambda GT11, lambda GT10, and a lambda ZAP. Okay, so the steps involved into the cDNA library construction. First, you have to synthesize the cDNA, and when you have to synthesize the cDNA, first you have to synthesize the first strand of the cDNA, then you have to synthesize the second strand of the cDNA, and after that, that cDNA you have to clone into the vector, and that cloning, it is a directional cloning, and to that directional cloning, it is going to be done into the vectors lambda GT10, lambda GT10, 11, and lambda ZAP. I think you may have cleared the idea about the cDNA libraries. If you like my video, please subscribe my channel and also share my video with your friends, so the knowledge, it is going to spread. Thank you.